Hello friends, welcome to the channel Fred Belly. This class is very useful for both CSIR as well as GATE and also some interviews. Here we will see the one of the major techniques for separation of protein. SDS-PAGE, native gel, isoelectric focusing, 2D polyacrylamide gel electrophoresis. These are the some of the techniques for separating protein or analysis of proteins. In this class, let us see what is the major thing about the first one, SDS page. This technique is very important. So, what this technique helps? This technique is going to help you to separate or analyze the proteins. The full form of SDS-PAGE is sodium dodecyl sulfate polyacrylamide gel electrophoresis. Sodium dodecyl sulfate polyacrylamide gel electrophoresis. So it is an anionic detergent that will literally act on the protein. It is going to denature the our protein. Also helps to visualize in better manner. So this SDS going to break down of protein into checks, and it is breakdown we can easily analyze them. These SDS page method is separate protein based on properties. These two are very important. One is mass. Mass is just nothing but weight. And another one is based on their size in our molecular biology. Now we will discuss the what are the reagents involved in this method. First one is SDS. We know that SDS is an anionic detergent. So, what is the charge of SDS? That is negative charge. That's why this type of reagent we cannot use in the DNA separation. So, SDS will move towards the anode. That's why we'll separate based on size only. SDS, which binds strongly to the denatured proteins to produce linear polypeptide chain. It binds strongly to and denatured proteins to produce linear polypeptide chains. SDS will be present for every two amino acids. It means in between two amino acids. Next reagent, that is beta mercaptanol or beta mercaptoethanol. What it will do? It is going to attach all these disulfide bridges. What it will do? It will attack all the disulfide bridges. For example, the protein X, single subunit protein, attack of beta mercaptoethanol, then what it will do? Breaks this type of disulfide bonds. Here the sulfide attached to amino acid breaks this type of disulfide bonds and gives SH. When we add the SDS, now the protein become negatively charged. Understood? If two separate proteins are present, protein X and protein Y, attached each other by disulfide bond, this is also same like that. Disulfide bond will break. After adding these two reagents, protein become negatively charged. So it is an anion now, and we know that it moves towards the anode. Got it? And so the separation is based on size of the particles. So charge is uniform. Okay. What is the principle of the gel? Polyacrylamide gel is cast as a separating gel topped by a stacking gel. This stacking gel has properties that cause the proteins in the sample to be concentrated into narrow band at the top of the separating gel. This polyacrylamide done by APS and TMAD, TMAD, TMAD. In agarose gel, it is a single gel, but in this polyacrylamide gel is single gel. It going to be two gels containing two parts. This is the polyacrylamide gel. Here it is a loading well, stacking gel. Normal it is 0.8 centimeter long. Below resolving gel, it is approximately 5 centimeter. Its pH 8.8, pH of stacking 6.8. First part is called stacking gel and the second one is called resolving gel. Let us take stacking gel. In this, all the proteins can easily run down because of they have large pore size. The purpose of this stacking gel is to concentrate the protein samples into sharp band before it enters to the main separating gel. Low concentration acrylamide gives large pore size, so which allows the to move freely. In case of resolving gel, have small pore size and resolving gel having pH 8.8 is higher than the stacking gel, that is pH 6.8. Resolving gel also it's called separating gel. Stacking principle. Due to the stacking have pH 6.8 and large pore size. Example, this is protein A, protein B moves as a same speed. This is gel A, protein B, stacking gel moves in same speed. Electrophoresis buffer containing glycine. These are the negatively charged anions. Glycine at pH 6.8. This is make it migration towards the anode, and also we tris chloride buffer. So, if higher pH is there, glycine moves very fast. Chloride ions moves slowly. So, when the current is switched on, in stacking gel, chloride ions having higher mobility than glycine. But SDS proteins have same speed. When it enters to the separating gel, due to the at pH 8.8, it is higher, the mobility of glycine is faster than the chloride ions. But SDS proteins separate based on their size due to small pore size. Understood? For visualization, we use loading buffer, bromophenol blue, and glycerol. Bromophenol blue and glycerol. What is the use? Glycerol increases the density of loading buffer. The sample can settle in loading well. Bromophenol blue is a colored dye and indicates the progression of electrophoresis. Thus, gel after electrophoresis has to be stained using protein specific dye, that is Comercia dye. Detection limit 200 nanogram. What it will do? The Comercia binds to the protein through ionic interactions between sulfonic acid groups and amino groups of the basic amino acids, as well as through van der Waals interactions between sulfonic acid groups and amino groups of basic amino acids. And we can also use silver nitrate. Silver nitrate. It brown in color, but Comercia gives the blue color. That's why we commonly use Comercia dye. Next, advantages of SDS-PAGE. SDS-PAGE is particularly useful for monitoring the protein purification procedure. As in this method, separating of proteins is based on the size. The relative molecular mass of the protein can also be determined by comparing with the standard proteins of known molecular mass. Since only sub-microgram amount of proteins are for the gel, very little materials is used in this form of purity assessment. Okay, friends, thank you.